Hello everyone, welcome to the course on medical biomaterials. Now we are going to talk about biofilm. Biofilm is a very important concept in biomaterial, and it affects quite a lot of biomaterial design and life of a biomaterial. What is this biofilm? The definition is it is a population of microorganisms concentrated at a solid liquid interface, okay, and surrounded by an extracellular polymeric substance, okay, it is called EPS matrix. So, this biofilm is attachment on material surface. It could be a stainless steel, or it could be a polymer, it could be a ceramic, it could be even the inside lining of vascular graft, okay. It may contain live cells, it may contain dead cells. It will contain proteins, sugars, polysaccharides, metabolites, that is metabolites that are produced by the bacteria, secondary metabolites, and also quorum sensing signaling molecules. So, a biofilm might contain lot of these, okay, and they are attached. At the early stages, the attachment is reversible, that means the material leaves, but as time progresses the attachment becomes more and more difficult, okay. Both gram negative and gram positive bacteria forms biofilms on indwelling medical devices. That means like I said, it could be a short duration device, or it could be a long duration device. Biofilm can form. Even in the urinary catheters, the first few hours bacteria can start accumulating, which is reversible, but as time progresses the attachment becomes very strong and the bacterial biofilm starts growing. So, large number of bacteria can form this biofilm. These are the common bacteria found like E. coli, Pseudomonas, Staphylococcus aureus, Staphylococcus epidermis, Proteus mirabilis, Klebsiella pneumonia, Enterococcus, Streptococcus, and so on actually. So, all these are bacteria found in biofilm, whether it is dental implant, whether it is urinary catheters, urethral catheters, and so on actually. All these bacteria can form biofilms. I just want to show some pictures. You can see the bacterial attachment and growth in some of these dental area. Look at this fracture plate, okay. There is an infection in that, you can see this, where the bacteria has settled down and formed biofilm, and it is more difficult to get rid of these type of bacteria and compared to bacteria which is in the sessile form or floating form. It has been found that almost you need 10 times more concentration of antibiotics to kill bacteria which are well established on surfaces. Like I said, it could be a metal surface, it could be a polymer surface, it could be a ceramic, and so on. Some more pictures, look at this. This is a prosthetic valve, you can see a lot of biofilm growth. Look at this, this is an urethral stent, okay. This is called a double JJ pig tail urethral stent. You can see bacterial infection. Generally in the urinary region you may have bacteria like E. coli or Proteus mirabilis there. So, biofilm is very common in medical implants. It could be even within a few hours, and it could last for years and years. So, addressing that biofilm is a very big issue in biomaterial research, because these biofilm can lead to infection, infection can lead to immune compromising or failure of the biomaterial, and so on actually. There are different strategies that are being practiced in getting rid of biofilm. Some of them are successful, some of them still needs a lot of research, and you cannot have one single strategy to get rid of all the biofilms, because depending upon where the material is introduced, the type, the nature of the bacteria and the biofilm could vary quite a lot. These are some more biofilm pictures. These are scanning electron pictures of Staphylococcus biofilm on polyurethane surface. You can see this, and this is Pseudomonas aeruginosa biofilm on polyurethane surface. Okay. So, you can see not only the bacteria, the exopolysaccharide, so it forms a thick layer. Okay. So, lot of materials, like I said, in different environments end up having biofilm. For example, if you look at catheters, central venous catheters made up of polyurethane, environment is blood, but still you can have Staphylococcus epidermis or Staphylococcus aureus or Pseudomonas Klebsiella type of biofilm. Look at hemodialysis catheters. Polytetrafluoroethylene is used here. Again the environment is blood, again Staphylococcus aureus or gram-negative anaerobes. Polyurethane is used in pulmonary artery catheters, again the environment is blood, you can have coagulase negative staphylococcus or enterococcus, pseudomonas, actinobacter. Urinary catheters like silicone or polyurethane or silicone, this is in the urine environment. You can have E. coli, you can have enterococcus, you can have protease metabolism. Look at peritoneal dialysis, silicone, it will have interaction with blood and fluids, solids, you can have Staphylococcus epidermis and Staphylococcus aureus. Enteral feeding tubes like PVC or polyurethane, generally fluids, again you can look at the type of bacteria here. Gastrostomy tubes made up of silicone, polyurethane, intestinal fluids, again Enterococcus, Staphylococcus, E. coli. Again endotracheal tubes, PVC, silicone, stainless steel, again you can have Staphylococcus, tracheotomy tubes, these are made up of PVC and silicone, generally its environment is air, you can have Staphylococcus epidermis and so on. So you can see the titanium, stainless steel in spinal, it is in contact with fluids, you can have a coagulase negative Staphylococcus or Mycobacterium tuberculosis. Penile implant silicone, it is in touch with urine and fluids, gram positive rods, cocci. Breast implants, silicones, polyurethane, you can have again coagulants negative Staphylococcus. Orthopedic, whether it is knee implant or hip or dental, defibrillators, different types of materials we are talking about, mostly metals, cobalt, chromium, aluminum, stainless steel, gold, and then inorganic material like zirconia, aluminum oxide. So they all have different types of bacterial contamination like Staphylococcus aureus, coagulase negative staphylococci, gram negative anaerobes and so on actually. And finally devices, different types of devices, biliary stent, intrauterine device like copper, vascular grafts, coronary stents, intraocular lenses, they all can end up having Staphylococcus aureus and epidermis and E. coli. So these are the common bacteria which goes, and as I showed you in the past 2-3 slides, the environment could be blood, there could be urine, it could be fluids, different types of fluids, cranial fluids, cerebrospinal fluids and so on. So we have metals, non-metals, polymers, and ceramics, they all have these type of infection. So what are the possible entry points of infection? Periodontal disease, something related to the teeth. Catheters, patients many times have urinary catheters inserted, so infection can happen. Implant surgery, a device is placed, whether it is an orthopedic, whether it is a dental, whether it is a knee, whether it is a stent, urethral stent or cardiovascular stent. Open wound, if there is a wound which is open, a lot of bacteria, Staphylococcus related bacteria can enter. So these are the points of entry, and it has been found that most of the infection happens because of the implant surgery, and almost 60 to 70% of rejection in the early days of implant is because of infection. So if you can address the implant related infection in the first 2 to 3 weeks, then almost 70% of the rejection of the implant could be overcome. So let us look at this biofilm development and dynamics. So what happens is bacteria, when it undergoes starvation, they can shrink and become spores. So they become very, very small, ultra micro bacteria, so they can attach to surface or they cannot be killed by antibiotics. So the attaching to surface, once they attach, there is a change in gene expression from swimmers to biofilm formers. So there is a change in gene expression of those bacteria when it is in the sessile form as against when it is in the biofilm form. They can encase themselves with a slimy matrix, we call it exopolysaccharide, a lot of sugar, so there is a slimy matrix that is forming on top of it. They because of the reduced nutrient availability for bacteria, okay, which are at the lower level of the biofilm, nutrition diffusion also slows down. So, the bacteria stops growing and they reach this type of starvation condition, and hence there could be change in the gene expression of that. Exchange molecular signal. Bacteria which are forming biofilms, they produce a chemical called quorum sensing, and thereby they can change their gene expression, and hence they can move from the sessile to the colonizer, and the bacteria can also identify that they are a large number of population which are in the colonizing stage. Chemical gradients, there could be, because of the biofilm which is a matrix, there could be a gradient which can create microenvironments for different microbial species with different activities to be present. So, the bacteria which are on the top of the biofilm, which gets nutrients, which get oxygen, which gets chemicals, may have a different environment. So, they will have a different activity and growth pattern when compared to bacteria which are right at the bottom of the biofilm, which do not get enough nutrient, which starve much longer, which do not get enough oxygen. Okay. So, antimicrobial drugs, antibiotics damage bacteria in the upper layer, but do not penetrate or present at low concentrations at the bottom. Okay. Okay. So, they are below the MICs when compared to the concentrations of antimicrobials available at the top of these bacteria. So, the bacteria which are at the bottom, okay, what happens is they become resistant to strains because they are getting drug concentration much below their MIC. So, shear forces can detach cells. So, there are cells which are attached on the surface, there could be flow, shear forces, blood flow or urine flow. It can detach the cells, and they can go out and again attach somewhere else and again form a protective layer. So, you can see there are so many things happening. It is a very dynamic process. The biofilm growth, maturation, detachment is a very dynamic process, and it is a very serious issue, especially in the area of biomedical device. So, there are two phases of pathogenesis of biomaterial centered infection: physicochemical interaction between bacteria and surface, molecular and cellular interaction. So, first is the physicochemical interaction between bacteria and surface, other one is the molecular and cellular interaction. Let us look at it. So, initially bacteria in the Brownian motion, they are moving around freely, they are in the sessile form. So, they start getting attached because of non-bonded interaction like van der Waals forces, gravitational forces, electrostatic forces, hydrophobic interactions, hydrogen bonds, and so on. Okay. So, they attach either on uncoated surface, or there could be already a protein layer which is formed on which these bacteria can be attaching. So, either on uncoated or on a protein coated. So, the cells deposit, absorbed, and slowly they get multiplied. Exopolysaccharides are formed. So, they start forming a big layer of exopolysaccharide, it is called the matrix here. Okay. So, the bacteria which are on the surface get enough oxygen and nutrient. The bacteria right at the bottom do not get as much because of the limitations of diffusion of both oxygen, nutrients, and other chemicals. So, they undergo genetic changes when compared to them, and the antibiotic flow is also controlled because of diffusion. So, the antibiotic may kill bacteria right at the top, whereas the concentration of antibiotic at the bottom may be so little, the bacteria here are not killed. So, they get exposed to antibiotic concentration much below the minimum inhibitory concentration. So, they may start slowly developing resistance to antibiotics. Okay. So, there could be antibiotic resistance bacteria at these layers which have changed their gene pattern, which have started growing slowly, and so on. Okay. Now, because of shear forces, because of the thickness of the surface of the biofilm, some bacteria may detach itself and start flowing downstream, and again this process of biofilm formation can keep happening. Okay. This is the process of biofilm. This can start in hours, and this can go right up to days. Okay. And some of these mature biofilm can be in weeks and go on forever and ever. Now, bacterial movement due to chemoattractants, the chemoattractants could be amino acids, sugar, oligopeptides. Okay. So, this concentration gradient it could be chemotaxis or hepotaxis. Okay. Chemotaxis, okay, which is diffusible. So, the concentration gradient happens because of the diffusion of these chemoattractants, whereas there are some attractants which are bound to the surface. So, the physical interactions are long range interactions, they are not very specific. It could be greater than 50 nanometer between the cells and surface, whereas short range interactions will be less than 5 nanometers, hydrogen bonding, ionic dipole interactions, hydrophobic interactions, these are all short range. So, these are all long range. So, we have two types of gradients produced because of chemotaxis and hepatotaxis. Okay. That is why the bacteria starts moving towards surfaces. The molecular and cellular interactions between bacteria and surfaces, there are many proteins on the surface of the bacteria, okay, which may be doing this type of job. So, the PSA, this is a galactose rich capsular polysaccharide adenosine composed of beta 1,6 linked N-acetyl glycosamines residues with some oxygen linked substituents of succinate, phosphate and acetate. Okay. Then we have the SAA, this is a slime associated antigen composed of N-acetyl glucosamine. We have PIA, this is a polysaccharide composed of beta 1,6 linked N-acetyl glucosamines with partly deacetylate residues. Then we also have AAP, this is an accumulation associated protein. So, we see the proteins on the surface of the bacteria and the proteins on the surface of the material surface, there is an attraction. So, the bacteria may have a lot of NH2 groups proteins, and the surface proteins may have a lot of SO3 groups. So, there could be attraction. So, this is the molecular and cellular interactions between bacteria and surface, whereas the physicochemical interactions could be because of presence of chemotaxis, because of non-bonded interactions and so on. Okay. So, PSA, SA take part in the bacterial material interactions, okay, whereas PIA and AAP in the cell-cell interaction. Okay. So, PIA is a polysaccharide composed of beta 1,6 linked N-acetyl glucosamines, whereas AAP is an accumulation associated protein. So, that leads into interaction between cell to cell, whereas PSA and SAA leads to interaction between the cells to surfaces. Okay. So, look at the biofilm mechanism, there could be environmental factors, there could be toxins, this could be cell growth process, and there could be something related to the surface proteins. So, environmental factors could be temperature, stress or starvation, because like I said, bacteria at the bottom of the biofilm are almost under starvation condition. Okay. So, bacteria is at sessile form, it moves into biofilm state. Okay. And there could be a reversible process also, biofilm dispersion because of enzymes like proteases which breaks the bond between the bacteria and the surface. We also have something called beta type phenol soluble modulins. These are supposed to be very toxins produced during the biofilm formation, which can lead into a lot of infection. Now, phase of the cell growth, so the bacteria grows and sometimes it reaches into stationary phase, okay, and inside the biofilm they may be in the stationary phase. Okay. Then we also have the beta toxins, so which are involved in the cell autolysis through eDNA, which is also a reversible process. Okay. So, all these are involved in the mechanism of biofilm formation. So, what are the problems associated with bacterial biofilms? Lot of clinical challenges, chronic or recurrent infection. A patient is a biomaterial is placed inside the patient and the biomaterial gets infected. So, the patient gets into chronic infection or recurrent infection, he is given antibiotics, it does not get cured, they are given stronger antibiotics and so on actually. Okay. Chronic inflammation, infection later on there could be inflammation, inflammation in the surrounding tissues, rapidly occurring antibiotic resistance, because as I said, the bacteria which are attached to the surface inside the biofilm slowly start acquiring antibiotic resistance. So, they are not able to be killed, whereas bacteria which are on the surface get killed. Spread of infectious emboli. So, the infection starts spreading on remaining parts of the surfaces. Okay. So, all these are problems associated with the bacterial biofilm. So, for example, if you look at the frequency of occurrence, urinary tract infection, when you place urinary catheters, like 10 to 20% of infection is because of this biomaterial centered infection. Percutaneous cardiac catheters, short indwelling catheters, temporary pacemakers, we are talking in terms of almost 10% max. Subcutaneous like peritoneal dialysis catheters, cardiac pacemakers around 4%. Soft tissues, mammary tissues, intraocular lenses. Circulatory system like prosthetic heart valves, vascular grafts, artificial heart. Okay. Bones, total knee replacement around 4%, prosthetic hip 2 to 4%. So, if you are thinking about having a long term indwelling biomaterial like hip prosthesis or knee replacement or cardiovascular stents or diaphragm valves, then long term use of biomaterials, okay, medical devices, you have to address this infection, and it is the frequent cause of failure. Okay. These numbers were obtained from this particular reference. So, what are the factors influencing bacterial adhesion? Environment. If there is continuous flow or if there is a stagnant flow. If there is a continuous flow, shear forces can prevent biofilm formation, whereas if there is a stagnant, then this can lead into a biofilm attachment. Material surface characterization like surface chemical composition, surface roughness, hydrophilicity, hydrophobicity, so on. Bacterial characteristic, bacterial hydrophobicity, hydrophilicity, bacterial surface charge. So, all these three factors affect the biofilm and bacterial adhesion. So, if you look at serum or tissue proteins, there are many proteins like fibronectin, which promotes Staphylococcus adhesion to the substratum surface. We have albumin, which inhibits bacterial adhesion to polymer, ceramic and metal surface, okay, because blood will contain all these proteins. By binding to the bacterial cells or changing the substrate surface to more hydrophilic. So, if you have albumin, it will inhibit bacterial adhesion, whereas if you may have fibronectin, it promotes Staphylococcus aureus adhesion, because albumin is very hydrophilic. So, when the surface becomes hydrophilic, then bacterial adhesion is reduced. Fibrinogen, it promotes adherence of bacteria, especially Staphylococci to biomaterial. So, fibrinogen also promotes. Thrombin, it also increases bacterial adhesion, it polymerizes fibrinogen in PPP to fibrin, because fibrin strands surrounds and link the platelet aggregate to stabilize the thrombus, which also promotes bacterial adhesion. Then we have this PPP, poor platelet plasma serum, the addition of various coagulant negative staphylococcus onto plasma coated material is much lower than onto the untreated control surfaces. So, generally many of these serum proteins except albumin, okay, increases bacterial adhesion like fibronectin, fibrinogen, thrombin. Okay. So, what is this? So, this effect is due to albumin, while IgG and FN are less effective, and due to Roman effect in which fibrinogen can be displaced by other proteins present in plasma such as high molecular weight. However, PPP with thrombin increases bacterial adhesion, because thrombin enhances bacterial adhesion, fibrinogen enhances bacterial adhesion, fibronectin also enhances bacterial adhesion. Now, so platelets increases Staphylococcus adhesion in comparison to human serum albumin, especially in combination with PPP and thrombin. Okay. So, platelets also increases Staphylococcus. So, as I said, generally PPP serum and albumin inhibits, whereas all other proteins in the serum sort of enhances in different ways the bacterial adhesion. Now, you may ask, what is this fibronectin? Okay, it is a 440 kilodalton glycoprotein of the extracellular matrix that binds to membrane spanning receptor proteins called integrins. Okay. Similarly, if you look at what is albumin, serum albumin is the main protein of human blood plasma and it is water soluble. What is this fibrinogen? It is also a protein produced by the liver and it helps in the blood clotting. Thrombin, it is a serum protease that converts fibrinogen into fibrin in blood clot coagulation actually. So, so serum also plays a very important part in addition of bacteria to material surfaces. Okay, so if you are having implants which are exposed to blood, then you have to consider these aspects also, because the blood protein, serum protein contains, tissue proteins contain a lot of different types which may enhance or retard bacterial adhesion to various material surfaces. Okay. There are some bacteria which become persistent once they form biofilms. Okay. So, these persistents or persisters, okay, these are a small subpopulation of bacterial cells that are dormant and extremely tolerant to antibiotics, because you have a thick extracellular polymeric surface that is formed on the material with the bacteria, live and dead cells. Antibiotics penetration becomes more problematic. So, the antibiotics right at the bottom of the biofilm concentration is so low, much below their minimum inhibitory concentration. So, those bacteria become tolerant to antibiotics. They also become persister, they are called persister cells. So, these persistence, it is a phenotype expressed by almost all bacteria including major pathogens. This can lead to chronic and relapsing infection. These persister cells can also tolerate high doses of bactericidal antibiotics. Small fraction of bacterial population responds and others form resistance or tolerance towards antimicrobial agents. So, that is one of the main reasons in bacteria, in biomaterial related infection, because of the formation of these persistent cells. Okay. We will continue about persistence of bacteria in the next class in more detail. Thank you very much for your time.